Welcome back. In the previous video, we have discussed about the structure of the genetic material, that is RNA, and in detail about the structure of the DNA. Now, the DNA molecule is quite long. So, if you have to calculate the length of a DNA molecule, let us say in a human cell. So, an average human cell has a size of around 100 micrometer. The smallest cell is the red blood cell. So, the average size is around 100 micrometer. And if you compare the size of the nucleus, it is roughly around 10 micrometer. So, it is 10% the size of the cell. Within the nucleus, we find the DNA in human cell as an example. So, the length of the DNA can be calculated simply by multiplying the total number of base pairs into the distance between the base pairs. We know that between one base pair and another base pair, the distance is 0.34 nanometer, that is 0.34 into 10 to the power minus 9 meter. This is what we discussed in the previous video. And the total base pairs in every human cell, as you know, is 6.6 into 10 to the power 9 base pairs. So, total number of base pairs into the distance, if you do, we get the total length of the DNA. So, that length is 6.6 into 0.34 is approximately 2 meters. So, you can imagine the length of the DNA is approximately 2 meters. This 2 meter long DNA molecule has to fit inside a nucleus, that is 10 micrometer. And if you consider inside the nucleus, we also have the nucleolus. The nucleolus will also occupy considerable amount of space. In the left space, the DNA molecule should be accommodated. So it is a huge task to accommodate such a long DNA molecule. Hence, we find that in organisms, the DNA is packed so that it can be fit inside the nucleus. In the case of prokaryotes, that is bacteria, we know that in a bacterial cell there is no nucleus and nuclear membrane is also absent. So, what we find is that the double-stranded DNA molecule is arranged somewhere in the center of the nucleus, and this area is what we call it as the nucleoid. So, this arrangement is done with the help of certain proteins. So, the DNA is negatively charged due to the presence of phosphate group, as you already know. Now, because of the presence of certain basic proteins which are positively charged, the DNA is arranged in a region called as the nucleoid region. So, certain proteins are involved in this, and also certain nucleoid associated proteins help in the organization of the DNA molecule. Bacteria don't have histone proteins because eukaryotes have histone proteins which will help in this organization. Exceptional cases, archaebacteria. Certain archaebacteria do have histone proteins which will interact with the negatively charged DNA and organize it in the region called as nucleoid. Coming to the arrangement in eukaryotes. In eukaryotes, the arrangement is quite complex. Say for example, the human cell. As you already know, the human cell DNA length is 2 meter long and the DNA is negatively charged due to the presence of phosphate group. Here in eukaryotes, they have special proteins called as histone proteins which will help in packing the DNA closer. So these histone proteins are basic in nature, and we know that proteins are made up of amino acids. Whatever be the charge of the amino acid, that would be the total charge on the protein. If a protein contains more of acidic amino acids, it will be negatively charged. If a protein contains more of basic amino acids, say lysine, arginine, in this particular case, it will give you a overall positive charge to the histone proteins. These positively charged histone proteins will combine with the negatively charged DNA helix, and together it will result in a structure called as the nucleosome. So to explain the packaging of DNA inside a human cell, we can use the nucleosomal model of packaging. What is this nucleosomal model? Let us understand this. Now, let us say this is the length of the DNA which needs to be packed, which has a negative charge throughout. Now, the positively charged histone proteins are of 4 types. So, histone proteins are H1, H2A, H2B, H3 and H4. So 5 different types of histone proteins will help in organizing the DNA molecule. So among these 5 histone proteins, H2A, H2B, H3 and H4, they come together and they form what is called as an octamer. So H2A, it contains 2 units, H2B contains 2 units, H3 contains 2 units and H4 contains 2 units. Total, these 4 histone proteins, they form what is called as an octamer, and this is the core of the histone protein. Around this histone core or the octamer, the negatively charged DNA molecule, it wraps around and it forms one complete turn and it comes out, and after some distance there is another nucleosome containing another octamer. It will wrap around this core and move. So that is how the DNA is wound around the histone octamer. The 5th protein, that is histone H1 protein, its role is basically to control the length of the DNA that is wrapped. So it acts as a valve in controlling the length of the DNA. So this is your H1 protein. So this is the structure of the nucleosome, where the DNA molecule is wrapped around the octamer core. Between the two nucleosomes, the length of DNA that is remaining is called as the linker DNA. As the name itself suggests, this DNA will link one nucleosome to another nucleosome. If you look at the size of the nucleosome, the rough size of the nucleosome is around, that is the diameter of it is around 10 to 11 nanometer, and the length of DNA that is wrapped around the octamer core, it is around 146 base pairs, and this length of the linker DNA, it can vary from 20 base pairs to 60 base pairs. So in general, a nucleosome structure is approximately 200 base pairs in length and 10 nanometer in diameter. That is the overall structure of the nucleosome. So this will give the DNA what is called as a bead on string appearance. So it looks as if there is a string on which beads are present at equal distances from each other. That is how the DNA looks like. So this is what is called as the chromatin, the thread-like structure that we see inside a nucleus. The thread-like structure, that is what is called as the chromatin. Chromatin is nothing but the nucleosomes that are present at equal intervals. This chromatin thread will then undergo more coiling to give a structure called as the chromatin fiber. This structure would be roughly 30 nanometer in length. This would further cause more coiling to give you a more compact size of 300 nanometer, which ultimately coils and produces a chromosome whose size is roughly 1400 nanometer. So that is how the DNA is packed inside the nucleus. So the negatively charged double helix DNA, it is wrapped around a histone core octamer whose length is around 146 base pairs. This length is controlled by the H1 protein. In between two beads or two nucleosomes, there is a linker DNA whose length is 20 to 60 base pairs. Each nucleosome has a diameter of roughly 10 or 11 nanometer. This size is fixed. This is what is called as a chromatin, and this chromatin will further undergo coiling to give you a structure whose diameter is 30 nanometer, giving rise to the chromatin fiber. The chromatin fibers will join and give you finally the chromosomes. These chromosomes we can see during the prophase stage, and very clearly we can see it during the metaphase stage. The chromatins will become furthermore coiled. So for higher level of coiling at the chromatin fiber stage and at the chromosome stage, there are other proteins called as the non-histone chromosomal proteins, which are required for further coiling of the DNA molecule. Non-histone chromosomal proteins, and that is how the 2-meter long DNA molecule is coiled into chromosomes whose size is roughly 1400 nanometer, which is present inside the nucleus. Now, when the chromatin molecule is coiling in a chromosome, so what we see is that in some regions the coiling is very high, and at some regions the coiling is very less. So what we see is that there is a dark region and there is a light region in the chromatin. The dark region, where there is high coiling of the chromatin, is called as heterochromatin. So here the chromatin is highly coiled, and this region appears to be dark in color when observed under the microscope, whereas the region where the coiling is less, so less coiling is seen, it appears to be light in color under the microscope. That region is called as the euchromatin region. Functionally speaking, the heterochromatic region, it is transcriptionally inactive, means it contains hardly few genes. Transcriptionally inactive region is the chromatin region. We can say that there are no genes at all. Whatever genes are present in a DNA is present in the euchromatin region, which is the region that is transcriptionally active. That is, the genes present in this region, they undergo transcription, giving rise to RNA, and these RNA molecules on translation give rise to the respective proteins. So that is how information is passed from the DNA to the RNA and RNA to the proteins with the help of the euchromatin region of a DNA. So this is an important question, difference between heterochromatin and euchromatin. Also explain the nucleosomal model of DNA with the help of a neat labeled diagram is a 3 or 5 mark question. So that is about the topic packaging of DNA helix in a human cell or in general in eukaryotes. Thank you.